Hello, this is Rubab, your audio navigator. The topic which I'm going to tell you about is cell biology, which is again a very huge topic, and you need to be aware of the very minute details. Questions generally asked are about 1 to 2 marks. They could be about the membrane structure or the transport or signal transduction cascades. So you need to cover each and every topic in it. Let's start with cell structure and organelles. First of all, you should know what is a cell and what is cell theory. What are the facts related to this theory? Then what are the different types of cells? They could be prokaryotic or eukaryotic. In prokaryotic, they could be bacteria or archaebacteria, or the eukaryotic consists of protista, fungi, plants and animal cells. Then you should know what is the structure of cells, what are the different parts of cell like cell wall, cell membrane, cytoplasm, nucleus, nuclear membrane, nucleolus, chromosomes, endoplasmic reticulum, ribosome, mitochondria, Golgi bodies, lysosomes, vacuoles and chloroplast. So you need to know the structure of these organelles and their functions. Then what is the prokaryotic cell structure? What are the general features for a prokaryotic cell? Then what are the structure and functions of a prokaryotic cell? It is comprised of it could be comprised of capsule, cell wall, cell membrane, cytoplasm, nucleoid, ribosome, plasmids, flagella or pili. Then what are the function of all these organelles and their functions? After that, what are the general features of eukaryotic cell structure? Then what is the structure of eukaryotic cell comprised of? Plasma membrane, nucleus, nuclear membrane, nucleolus, mitochondria, endoplasmic reticulum, ribosome, Golgi bodies, lysosomes, cytoplasm and chromosomes. So go through all these cell structures and their functions. Then how the plant cell structure is different from the animal cell structure. They are different as they have cell wall, chloroplast, vacuoles, etc. As for the organelles are concerned, they could be divided into membrane bound organelles and non-membrane bound organelles. So you should know the examples of both of these. So for this section, you need to study the structure and function of different organelles and how each cell is different from the other. For example, plant cell and animal cells and prokaryotic and eukaryotic cells. Now you need to study about the biological membranes. First of all, their structure and dynamics. So first of all, you should know what are the common features of the diverse range of biological membranes. Then you need to study the structure. What are they mainly comprised of? Lipids and proteins. Phospholipids are the major class of membrane lipids. And the other type of membrane lipids are the glycolipids and the cholesterol. So you should know the fatty acid chains in phospholipids and glycolipids usually contain an even number of carbon atoms in the membrane, typically between 14 and 24. And the most common ones are the 16 and 18 carbon fatty acids. And also go through what are the other features of these membrane lipids. For example, these membrane lipids are amphipathic molecules. That means they contain a hydrophilic and a hydrophobic moiety. These phospholipids and glycolipids readily form bimolecular sheets in aqueous medium. The lipid bilayers are non-covalent and have a cooperative structure. Then you should also need to go through the permeability of these lipid bilayers. They are highly impermeable to ions and most polar molecules. So the transport of these kind of molecules through the membrane is done through the channels or carriers. You should know that how the flow of ions through a single channel in a membrane can be detected, and what are the molecules which carry out most of the membrane processes. These are the proteins. The membrane proteins could be peripheral or integral, so you should know about them. What are the interactions holding them? Another important property of membrane lipids and proteins is that they diffuse rapidly in the plane of the membrane. It could be the lateral diffusion, which is very rapid, or it could be the transverse diffusion, which is slow. What is the fluid mosaic model of lipids and proteins in biological membranes? Then how are all biological membranes asymmetric? And how is the fluidity of membrane controlled? It is done by fatty acid composition and cholesterol content. So you need to go in detail how do they perform this function. Then where are the carbohydrate units located in the plasma membrane? They are located at the extracellular side of the plasma membranes. You also need to know that how the functional membrane systems can be reconstituted from purified components. So you should know how is it done. Then what is glycophorin, which is a transmembrane protein, forms a carbohydrate coat around the red cells. So you should go through about its structure and how is it embedded in the membrane. Then how the transmembrane helices can be accurately predicted from the amino acid sequences. This is a very important point. You should know about it. What are the amino acid sequences responsible for the transmembrane property of proteins? Then what is spectrin, which is a membrane cytoskeleton that makes erythrocytes resistant to shear. So you should know about its structure. After this, you need to study how the transport is done across the membranes. You should know what is the major difference between channels and pumps. For example, channels enable ions to flow rapidly through membranes in a thermodynamically downhill direction, while the pumps use a source of free energy such as ATP or light to drive the uphill transport of ions or molecules. So you need to study a few examples of these. For example, acetylcholine receptor, which is a channel that mediates the transmission of nerve signals across synapses. So you need to go through the detailed function of this receptor and also the structure, and also how does the patch clamp conductance measurements reveal the activities of single channels. Just go through this. How is it done? Then how does the binding of two acetylcholine molecules transiently opens a cation selective pore? Let's go through this in detail and all the facts which are related to this. After that, you need to know how do the voltage gated channels work, how a nerve impulse in an electric signal produced by the flow of ions across the plasma membrane of a neuron. The action potentials are mediated by transient changes in sodium potassium ion permeability. So you need to go through this in detail. So what is the effect of few potent neurotoxins like tetrodotoxin and saxitoxin? They block sodium channels. Then how does the 4 repeating units of the sodium channel form a highly discriminating pore? Then after that, how does the potassium channels work? You need to go through this. They are highly selective. What is the ball and chain model for the inactivation occurring by the occlusion of the pore? What are gap junctions? They allow ions and small molecules to flow between communicating cells. So you should know how do they work. Then how does the active transport work and what does it require? How does the ATP hydrolysis drives the pumping of sodium and potassium ions across the plasma membrane? You should know how much energy is required. For example, 3 sodium and 2 potassium ions are transported per ATP hydrolysis in the unidirectional cycle. Then how does the binding cavity alternatively faces the inside and outside of the cell in each transport cycle? So you need to know the mechanism. Then what is the role of digitalis? How does it inhibit the sodium potassium pump? By blocking its dephosphorylation. Then after that, how does the calcium ion are pumped out of the cytosol? Then you also need to go through that how the sodium gradient across the plasma membrane could be tapped to pump ions and molecules. So you should know how does it happen. Then what are the many bacterial transport processes which are driven by the proton flow across the cell membrane? So you need to go through different examples. For example, bacteria rhodopsin is a light-driven proton pump in halobacteria. So you need to know the mechanism, how does it work. So after this, the next topic which you need to cover is the signal transduction. So what are the different signal transduction cascades which are used for sensing and processing of stimuli? They are constructed by receptors, enzymes, channels and regulatory proteins. So we need to know the examples and the mechanism of how do they work. So start with the chemoreceptors on bacteria which detect attractants and repellents and send signals to flagella. Then how does the direction of flagellar rotation determines whether bacteria swim smoothly or tumble? There are 4 kinds of chemoreceptors which transmit signals across the plasma membrane, so you just go through them briefly. After that, what is the mechanism? How is the adaptation to chemotactic stimuli is mediated by reversible methylation of chemoreceptors? So you should know about the cascade and what are the enzymes involved in it. Then coming to the animal kingdom, how a retinal rod cell can be excited by a single photon. Then what is the structure of rhodopsin, which is the photoreceptor protein of rods. And how does the visual excitation is triggered? It is done by the photoisomerization of 11-cis-retinal. Then what is the cascade which is activated by the photoexcited rhodopsin? It is the G protein cascade which leads to the cyclic GMP hydrolysis. So you need to know the whole mechanism for it. And how does the cyclic GMP hydrolysis closes the cation specific channel to generate a nerve signal. Then how does the color vision is mediated? It is done by the 3 cone receptors that are homologs of rhodopsin. What is the role of cyclic AMP? It is a second messenger in the action of many hormones. And how is it formed? By adenylate cyclase. Then what is the mechanism of the stimulation by the cyclic AMP of the phosphorylation of many target proteins by protein kinase A? So what is the role of PK in different pathways and cascades? You need to know about them. Then what is the phosphorinositide cascade? How does it work? How does the inositol 1,4,5 trisphosphate opens channels to release calcium ion from intracellular stores? And what is the role of diacylglycerol? It activates protein kinase C, which phosphorylates many target proteins. So you need to know about them briefly. And you should know that the 7 helix receptors and G proteins play key roles in diverse signal transduction processes. So go through them. Different examples related to this. What is the role of calcium ion? How it is a ubiquitous cytosolic messenger. Then what are EF hands? What are their role and significance? Then how does the calcium bound form of calmodulin stimulates many enzymes and transporters? What are these transporters? Examples. Then also go through the mechanism of how the membrane spanning receptor tyrosine kinase control cell growth and differentiation. How the receptor tyrosine kinase is activated, and how the activated tyrosine kinase receptors are recognized by SH2 domains of target proteins. Then how do many cancer producing genes encode alter signal transduction proteins? What are the examples and how do they work? For example, mutations of RAS blocks its GTPase activity, leading to cancer. So what is the role of RAS, which has a key role in the control of cell fate? So you need to know about it. What is the mechanism of action? So these were the few signal transduction cascades. Go through them briefly, as it will be difficult for you to remember it thoroughly, but the concepts about them should be clear to you. Plus the role of few important proteins and molecules in the cascades. They are very important and could be asked in the exam. Then you need to go through about the hormones and neurotransmitters. What are these molecules and what are their roles? They are both the signaling molecules. Then how are they different from each other? Like what are the signals they carry? What are the pathways they undergo? Then what is the speed by which the information flows? What is the duration of effect? And what is the type of action and response? Then what are the target cells or molecules? So you need to go through all of them with examples. What are the different hormones which are present in a human body and what are their roles and function? From where are they secreted and how their secretion is controlled? What is the effect of them on human body? And what happens when malfunctioning of the hormones is there? So need to cover all these points in detail. Likewise, study about the neurotransmitters also. This ends the topic of cell biology. So go through all the points which I have discussed. They are important from the exam point of view. Generally, concept-based questions are asked from here. So all the concepts should be clear to you. Time which you need for its preparation is around 2 to 3 days. I hope the information which I shared was helpful to you, and I wish you good luck. Thank you.